Hi, I'm Sharanya. In this presentation, I'm going to talk about the presumptive and confirmatory blood tests for forensic analysis. What is serology? Serology is described as a range of laboratory tests utilizing antigen and serum antibody reactions. Forensic serologists analyzes blood, saliva, urine, semen samples. There are two different types of blood and body fluids transfer. One is direct transfer, the other one is indirect transfer. Current serological test analyzes the presence of biological fluids along with investigations. Screening of evidence usually includes visual examination, that is presumptive test, and serological examination, that is confirmatory test. Blood is a body fluid composed of plasma and serum. Blood contains red blood cells, white blood cells, and platelets. RBC or red blood cells contain hemoglobin, which is a 4 subunit of protein. For analyzing blood, there are two different presumptive tests available: Luminol Presumptive test and Phenolphthalein Presumptive detection test. Presumptive tests are safe, inexpensive, and easy to perform. So the Luminol Presumptive test is based on the addition of luminol reagent. The luminol reagent will react with the heme group present in hemoglobin, producing a fluorescence that is bluish white or yellowish gray. Areas can be rapidly evaluated by spraying it with luminol reagent and a luminescence is observed. In the picture here, there are two different pictures. One is before the addition of luminol reagent and the second picture is after the addition of luminol reagent, showing a fluorescence. The second test is phenolphthalein presumptive detection test, also known as Castle-Meyer test. And this Castle-Meyer test involves the addition of 1:5 solution of phenolphthalein and ethanol. Just add 1 or 2 drops, wait for few seconds, and add few drops of 3% hydrogen peroxide. The principle of Castle-Meyer test is peroxidase activity of heme group present in the blood. The hemoglobin acts as a catalyst for the oxidation of the colorless phenolphthalein to pink magenta in color. The picture above shows the positive result for phenolphthalein detection test. The quadrant swab applicator changes color to pink or magenta. Next is membrane immunochromatographic assay confirmatory test for blood. This membrane immunochromatographic assay test is also known as lateral flow immunoassay test. They are used in clinical microbiology labs and they have double antibody sandwich assays. This test have two different zones, capture zone and control zone. Capture zone is also known as test line. The capture zone contains immobilized antibodies. Lateral flow immunoassay test used for analyzing blood. They are used in clinical microbiology labs. They have two different zones present, capture zone or test line and control zone. The capture zone or test line contain immobilized antibodies. The specimen contain antigen is placed on a sample pad and they migrate into the conjugate pad where there are conjugated antibodies. As a result, antigen-antibody complex will be formed. This migrates towards the capture zone and become visible on the test membrane. The sample again migrates to the control zone and produces the second visible line. So when there is a positive result, a single line appears in both control zone and the capture zone. When there is a negative result, a single line appears only in the control zone, and when there is an invalid result, a single line in test area without a line in the control zone will be formed.